Hey there, curious minds. Welcome back to the Deep Dive, the show where we untangle complex information and transform it into clear, engaging insights. Today, we're embarking on a truly fascinating journey into one of the body's most dynamic and, well, essential processes, lipid metabolism. We're taking a deep dive into Section V of Mark's Basic Medical Biochemistry, specifically zeroing in on chapters 29, 30, and 31. Our mission today is basically to guide you step by step through the intricate dance of fats in your body. We'll explore everything from, the moment dietary lipids enter your system, how they're stored away, how they're rigorously burned for energy, and even how they're meticulously built into the crucial cellular structures that make you, you. We're not just reciting facts. We're going to connect all the biochemical pathways, look at some surprising clinical examples, and ensure you walk away with not just knowledge, but genuine aha moments about why fats are so incredibly critical. Think of this as your essential guide to truly understanding the profound significance of lipids and what truly happens when this finely tuned system goes, well, a little sideways. Let's get started. So we talk a lot about fats in our diet, but in the body, we usually refer to them as lipids. What exactly are we talking about when we say lipids, and why is their sheer diversity so important to grasp right from the beginning?
That's a great starting point. What's truly fascinating here is just how incredibly diverse lipids are. It goes way beyond just, fat. They're not just one thing. They fall into these broad categories like fatty acids, which are kind of the basic building blocks, right?
Okay.
And then triacylglycerols, those are probably the most recognizable as they're the major way we store fat for energy. But we also have glycerophospholipids and sphingolipids, which are absolutely vital structural components of all our cell membranes.
The cell walls themselves, basically.
Exactly, the membranes. Then there are eicosanoids, which act as these powerful local signaling molecules, cholesterol, which is a precursor for hormones and bile salts, and of course the fat-soluble vitamins A, D, E, K. Despite their incredibly varied structures and functions, everything from energy storage to sending cellular messages to literally making up the fabric of our cells, they all share one fundamental defining property: their relative insolubility in water.
Right, the oil and water thing.
Precisely. And this insolubility is the common thread, the big challenge that dictates how they're handled throughout the body. It drives the need for all these complex digestive and transport mechanisms we're about to unpack.
And before our bodies can truly use these diverse lipids, especially the big ones like triacylglycerols from our food, they need to be broken down. That initial breakdown starts surprisingly early, doesn't it? We have some initial players even before the small intestine gets involved.
Absolutely. The journey for dietary triacylglycerols, the main fat in our diet, actually begins right in the mouth with an enzyme called lingual lipase.
In the mouth.
Yes,
Yeah.
active even before you swallow. Then, in the stomach, gastric lipase takes over. These particular lipases are especially good at targeting triacylglycerols containing short and medium-chain fatty acids. While they don't break down all the fat, they sort of initiate some preliminary hydrolysis. They're particularly important for infants because their main dietary fat source, milk, is rich in these shorter fatty acids.
Okay. Makes sense.
For most of the dietary fats we consume, especially the longer chain ones, the bulk of the digestion still happens further along, down in the small intestine. So they're important starters, but maybe not the main event for a typical adult meal.
Okay, here's where it gets really interesting, because the real heavy lifting for fat digestion happens in the small intestine. But here's that puzzle again. Fats and water don't mix. So how does the body effectively break down these water-insoluble fats in the watery environment of our gut? This is where bile salts come in, right?
This raises a truly critical point in lipid metabolism.
Yeah.
Yeah, imagine trying to digest a big blob of oil just floating in water. It presents almost no surface area for enzymes to actually get to it. The answer is a process called emulsification, and bile salts are the body's really elegant solution.
Emulsification. Okay.
Bile salts are what we call amphipathic compounds. Think of them as molecular detergents, basically. They have both a hydrophobic part, water-fearing, and a hydrophilic part, water-loving.
Like soap.
Exactly like soap. They're synthesized in the liver from cholesterol and then stored and secreted by the gallbladder. When fats enter the small intestine, bile salts literally coat these large fat globules, breaking them down into tiny dispersed droplets. You can picture it just like vigorously shaking oil and vinegar to form a temporary emulsion in salad dressing. That's emulsification.
Right.
This process doesn't chemically digest the fats, but it's absolutely vital because it vastly increases the surface area. It makes it exponentially easier for the actual digestive enzymes to access their substrates and start working.
So without bile salt.
Without this crucial step, yeah, most of the fat you eat would simply pass right through you, undigested. It's fundamental.
Once those fat globules are beautifully emulsified, we need the enzymes to do the chemical cutting, the breaking down, and there's a sort of dynamic duo, specifically for triacylglycerols, isn't there, that really takes center stage?
Indeed. The major enzyme responsible for the bulk breakdown of dietary triacylglycerols is pancreatic lipase. It's highly efficient, very specific. It acts at positions 1 and 3 of the glycerol backbone to release 2 free fatty acids and a single 2-monoacylglycerol molecule.
Okay, specific spots.
Yep. And what's crucial to understand is that pancreatic lipase doesn't work alone in this challenging environment. It has a critical helper protein called pancreatic colipase. Colipase. It's a protein that binds to both the bile salt-coated fat droplet and to the lipase itself. It basically acts as an anchor.
Oh, interesting.
Yeah, it prevents the bile salts from kind of pushing the lipase off the lipid surface and allows the enzyme to function efficiently right there at the oil-water interface. Beyond triacylglycerols, other enzymes are also working away. Cholesterol esterase hydrolyzes cholesterol esters, and phospholipase A2 breaks down dietary phospholipids. It's a whole team effort, breaking down pretty much all major dietary lipids into forms we can absorb.
It sounds like a beautifully orchestrated process, like you said. And like any good orchestra, it needs a conductor to signal when and how to play. What signals get the gallbladder and pancreas to release these crucial digestive agents right when fats arrive?
Yeah, the timing is key. The hormones, cholecystokinin, or CCK, and secretin play these key regulatory roles. They're the conductors for this digestive symphony.
CCK and secretin, okay.
Cholecystokin is secreted by specialized cells in the duodenum and jejunum of the small intestine, specifically in response to the presence of fats and partially digested proteins. CCK then sends signals out. It tells the gallbladder to contract and release its stored bile into the small intestine.
Squeeze out the bile.
Exactly. And simultaneously, it stimulates the pancreas to secrete its full suite of digestive enzymes, including that pancreatic lipase and colipase we just talked about. Secretin, on the other hand, is released more in response to the acidic chyme entering the duodenum from the stomach.
The acidity triggers it.
Right. Its main job is to stimulate the pancreas to secrete a bicarbonate-rich fluid. This bicarbonate neutralizes the stomach acid, creating the optimal, slightly alkaline pH environment that's absolutely necessary for these pancreatic enzymes to work properly. Without that precise hormonal coordination, fat digestion would be really inefficient and potentially damaging due to the acid.
So the fats are now broken down into smaller units. Free fatty acids, 2-monoacylglycerol, cholesterol, etc.
Yeah.
How did these smaller lipid-soluble molecules actually get into our intestinal cells for absorption? They're still water-fearing, right?
This is another elegant solution to that solubility problem. They form what are called micelles.
Micelles. Okay, I've heard of those.
They're tiny microdroplets, just a few nanometers across. The bile salts arrange themselves with their hydrophobic parts facing inwards, basically hiding the fatty acids. 2-monoacylglycerol, cholesterol, and fat-soluble vitamins inside this core. Their hydrophilic parts face outwards, interacting with the watery environment.
So they can travel through the water layer.
Exactly. This micelle structure is absolutely key because it allows these lipid-soluble components to diffuse through what's called the unstirred water layer, a thin, watery barrier right at the surface of the intestinal lining, and then they can passively enter the enterocytes, the intestinal cells. It's like a tiny ferry service.
A ferry, I like that.
It's important to note an exception here, though. Short and medium-chain fatty acids, those with fewer than 12 carbons, are actually soluble enough to be directly absorbed into the chortle blood. They don't need micelles or the chylomicron packaging we'll talk about.
Ah, okay, so they take a shortcut.
Kind of, yeah.
Once inside these intestinal cells, these components don't just, hang out. They're quickly repackaged.
Yeah.
So let's unpack this. How do the intestinal cells rebuild triacylglycerols from these absorbed bits and pieces?
Yeah, inside the intestinal cells, there's this rapid and really efficient process to rebuild the fats. The absorbed free fatty acids are first activated. They get converted to fatty acetyl-CoA.
Activated? What does that mean?
It basically means attaching them to coenzyme A, which requires ATP. This activation makes the fatty acids chemically reactive, ready for the next step.
Got it. Energy input.
Right. Simultaneously, the 2-monoacylglycerol, that main product from pancreatic lipase, is efficiently converted back to diacylglycerol by adding one of those activated fatty acyl-CoAs, and then to triacylglycerol by adding a second one. This resynthesis pathway ensures the newly absorbed fatty acids are quickly re-esterified back into their storage form. It prevents them from accumulating as free fatty acids, which could actually be toxic to the cell.
So rebuild them quickly.
Exactly, like a busy assembly line putting the parts back together into a more stable, transportable package.
And all these newly synthesized lipids, along with the dietary cholesterol and fat-soluble vitamins that came in, are then packaged into a very special, large transport particle. What is this crucial package called, and what's inside it?
This is where chylomicrons are born.
Yeah.
And they are truly remarkable things, biologically speaking.
Chylomicrons.
They are the largest of the lipoprotein particles, essentially tiny lipid-rich cargo ships designed specifically to transport dietary fats from the intestine out to the body. Visualize a sphere with a core primarily made up of those newly synthesized triacylglycerols and cholesterol esters, all very hydrophobic. This core is surrounded by a monolayer of phospholipids, some free cholesterol, and a crucial protein component, apolipoprotein B48 or ApoB48.
ApoB48. That sounds important.
It is absolutely essential. Without ApoB48, these particles cannot be assembled or secreted from the intestinal cells. The body would basically be unable to absorb dietary fat properly.
Wow. And these nascent chylomicrons, as they're called initially, they're too big to go straight into the bloodstream like sugars or amino acids, right? They take a different route.
Precisely. Unlike most other absorbed nutrients that go directly into the capillaries and then the portal vein to the liver, chylomicrons are simply too large to squeeze into those tiny blood vessels in the intestine. Instead, they are secreted into the lymphatic system, the lacteals within the intestinal villi.
The lymph system. Interesting detour.
It is. They travel through the lymphatic vessels, which eventually merge and empty into the bloodstream via the thoracic duct, which connects near the heart, specifically into the subclavian vein. This is a really critical step because it ensures that dietary fats get distributed throughout the body to peripheral tissues like muscle and adipose tissue before they reach the liver.
Ah, so they bypass the liver initially.
Yes. If they went straight to the liver would process them too quickly, maybe hoard them, and other tissues wouldn't get their fair share of this crucial energy source right after a meal. It's an elegant way to deliver large amounts of fat globally.
And these nascent chylomicrons, fresh out of the intestine, aren't quite ready for prime time until they pick up some extra components in the bloodstream.
That's right. As these newly released nascent chylomicrons enter the bloodstream, they're not fully functional yet. They undergo a crucial maturation step. They interact with high-density lipoproteins, or HDL, which circulate in the blood and often act as a kind of apolipoprotein reservoir.
HDL, the good cholesterol particles.
The very same. From HDL, nascent chylomicrons acquire two additional very important apolipoproteins. Apolipoprotein CT, ApoCI, and apolipoprotein E, APE. These additions are absolutely crucial because they convert the nascent particle into a mature chylomicron, making it fully equipped and functional for efficient lipid delivery to the tissues that need them. ApoCI, for instance, is the key that activates the enzyme that will unload the fats.
Okay, so they need those extra proteins to do their job.
Exactly. Once mature, these chylomicrons efficiently travel through the circulation. If you were to draw blood after a particularly high-fat meal, you might actually see a transiently cloudy or creamy appearance to the plasma because of all these particles.
Wow.
Their primary role is delivering the huge amounts of triacylglycerols they carry to peripheral tissues that need energy or storage. Think cardiac muscle, skeletal muscle, and adipose tissue. This delivery is done by an enzyme called lipoprotein lipase, or LPL. It's anchored to the capillary endothelium, the inner lining of blood vessels in these target tissues.
LPL, okay, another key enzyme.
A very key enzyme. ApoCI on the chylomicron surface activates LPL. LPL then efficiently digests the triacylglycerols inside the chylomicrons, releasing free fatty acids right there at the tissue surface. These fatty acids are then readily taken up by the adjacent cells. Muscle cells burn them for energy. Adipose cells re-esterify them back into triacylglycerols for storage.
So the muscle cells can use those fatty acids for energy right away, and adipose tissue can store them up for later. Makes sense. What happens to the chylomicron itself once most of its fatty cargo has been offloaded? Does it just dissolve?
Not at all. As LPL actively strips away the triacylglycerols, the chylomicron particle gets progressively smaller and significantly denser. At this point, it usually sheds its ApoCI, returning it back to HDL, but crucially, it keeps its ApoB48 and ApoE. This transformed particle is now called a chylomicron remnant.
A remnant. Leftovers, basically.
Sort of, yeah, but important leftovers. These remnants are still rich in cholesterol esters because LPL doesn't really touch the cholesterol, and they essentially carry the remaining lipid cargo, particularly that dietary cholesterol, back towards the liver.
Ah, so the liver gets the cholesterol in the end.
Exactly. If we connect this to the bigger picture, these chylomicron remnants are then rapidly cleared from the circulation by the liver. The liver has specific receptors on its surface, including one called the LDL receptor-related protein, or LRP, that recognize the ApoE on the remnant surface. This recognition triggers their endocytosis. The liver cell engulfs the entire remnant particle.
So the liver cleans them up.
Precisely. This removal process is absolutely crucial. It prevents the buildup of these potentially harmful cholesterol-rich remnant particles in the blood, which could contribute to atherosclerosis over time. It also allows the liver to recycle or further process their remaining cholesterol and other components. It's an efficient cleanup mechanism, ensuring the bloodstream isn't overloaded with lipid particles long after a meal.
This whole system is so intricate, so many moving parts. And when any one of them falters, there can be pretty significant health consequences, right? It seems like a cascade of problems could happen. Let's look at a few examples from the book, starting with Will S. His story highlights a really common issue: gallstones.
Yes, Will S.'s case of acute cholecystitis gallbladder inflammation and the subsequent obstruction of his common bile duct directly impacts lipid digestion at a really fundamental level. The blockage prevents the normal flow of bile salts into the intestine. Remember how crucial they are for emulsification.
Right, the detergents.
Exactly. Without enough bile salts, the emulsification of dietary fats is severely compromised. This leads directly to their malabsorption. Clinically, this shows up as steatorrhea, that's the presence of excess fat in the stool. It's often described as bulky, glistening, maybe yellowish, and foul-smelling. Basically, the fat is just passing through undigested.
So you're not getting the energy from it.
You're not getting the energy, and it causes those unpleasant symptoms. The case also mentions elevated bilirubin levels, which is related to the blocked bile flow causes a backup in the liver's waste disposal, leading to jaundice. But the steatorrhea really underscores how essential bile salts are for fat processing.
Okay, another case mentioned is Al M., who suffered from alcohol-induced pancreatitis. This sounds like a different kind of enzyme problem.
Al M's situation vividly illustrates how inflammation of the pancreas, often triggered by things like chronic alcohol abuse, can lead to a severe reduction in the secretion of pancreatic digestive enzymes, including that key player, pancreatic lipase.
So the pancreas itself is damaged.
Right, the pancreas is inflamed, maybe even attacking itself. Without enough lipase being secreted, dietary triacylglycerols just cannot be properly hydrolyzed into absorbable fatty acids and 2-monoacylglycerol, no matter how well emulsified they are by bile salts. So again, this results in severe malabsorption and, predictably, steatorrhea. The elevated serum amylase and lipase levels mentioned in his diagnosis are key lab markers for pancreatitis, directly reflecting the inflammatory process spilling these enzymes into the blood instead of the gut. It's a powerful example of how inflammation in one organ can cripple a fundamental digestive process elsewhere.
Beyond these direct medical conditions, the text also mentions dietary factors that can mess with lipid absorption, like artificial fat substitutes or certain weight loss drugs.
Yes, that's right. Consider Olestra, an artificial fat substitute that was developed to reduce calorie intake from fat. Its unique structure, basically a sucrose molecule with 8 fatty acids attached, makes it impossible for a pancreatic lipase to hydrolyze it. It just doesn't fit the enzyme.
So it passes right through.
It passes through the digestive system completely unabsorbed, eliminating its caloric contribution. But this also means it can cause significant gastrointestinal side effects, like cramping and diarrhea. And maybe more importantly, because it doesn't get properly digested or absorbed, it can interfere with the formation of those micelles we talked about. This can impair the absorption of essential fat-soluble vitamins, A, D, E, K, which rely on micelles for uptake.
An unintended consequence.
Definitely. Similarly, the drug orlistat works by directly inhibiting pancreatic lipase. It's designed to reduce dietary fat absorption for weight loss, but predictably, it causes steatorrhea as a common side effect. Both examples serve as a stark reminder that interfering with one part of this complex system often has downstream effects.
And then there's these quite severe genetic conditions that deeply affect the process, like abetalipoproteinemia. That sounds like a fundamental problem with building those fat transport vehicles, the chylomicrons.
You're spot on. Abetalipoproteinemia is a severe, though rare, genetic disorder. It's caused by a deficiency in a crucial protein called microsomal triglyceride transfer protein, or MTP.
And DP.
This MTP protein is absolutely essential for loading triacylglycerols and other lipids onto apolipoprotein B48, ApoB48, to actually form chylomicrons in the intestine. It's also needed to load lipids onto ApoB100 to form VLDL in the liver, which we'll touch on later. Without functional MTP, these crucial lipoproteins simply cannot be assembled or secreted from the intestinal cells or the liver.
So the fat gets stuck inside the cells.
Exactly. It accumulates in the intestinal cells and liver cells. What this means for the patient is profound malabsorption of dietary fats and all the fat-soluble vitamins. This results in severe steatorrhea, major deficiencies in vitamins A and E, which can impact vision and nerve function, and a range of severe neurological issues over time. It really raises an important question, doesn't it? How many seemingly unrelated symptoms, vision problems, nerve damage, digestive issues, could potentially trace back to a single deficiency in one metabolic protein? It powerfully underscores the interconnectedness of these pathways and how a single protein can be a linchpin for global health and development.
Okay, so we spent a good amount of time talking about how we get fats into our bodies and transport it around. Now let's pivot and explore how our bodies actually use them for energy. When do fatty acids become a major sort of go-to fuel source for us?
Right, the utilization part. Fatty acids are indeed a major fuel for humans, providing a substantial amount of energy, especially during periods of increased demand or when other fuel sources like glucose are running low. For instance, right after a meal, when glucose is abundant, we tend to store excess fatty acids and carbohydrates as triacylglycerols in our adipose tissue. That's the storage phase.
Filling up the tanks.
Exactly. But during periods like an overnight fast, which can deplete our immediate glucose and glycogen stores, fatty acids become the primary energy source for many tissues. They also become crucial during prolonged, moderate-intensity exercise when muscle glycogen starts running low. Using fats allows us to sustain activity for much longer.
So basically, when glucose gets scarce, the body flips a switch and turns to these vast fat reserves, mobilizing fatty acids to meet its energy needs. It's an incredibly efficient way to store long-term energy. And which tissues in the body are particularly good at using these fatty acids for energy? Are there any that maybe struggle or have unique ways of dealing with it?
The cardiac muscle, the heart, which is constantly working and has a huge, unrelenting energy demand, is a major user of fatty acids. Skeletal muscle, especially during prolonged activity, also heavily relies on them. And the liver is a major site of fatty acid oxidation too, although it uses them differently, often converting them.
Okay, heart, muscle, liver. Big users.
Right. These tissues have a high capacity for fatty acid oxidation, meaning they're packed with mitochondria and all the necessary enzymes. Interestingly, the nervous system, particularly the brain, has a pretty limited ability to directly oxidize fatty acids.
Why is that?
It seems to be partly due to the blood-brain barrier restricting the entry of long-chain fatty acids bound to albumin and perhaps a lower level of some key enzymes. However, during prolonged fasting or starvation, the liver performs this incredible feat of metabolic adaptation. It starts converting fatty acids into ketone bodies, specifically acetoacetate and beta-hydroxybutyrate.
Ketone bodies. Okay, we hear about those with certain diets.
We do. These ketone bodies are water-soluble, unlike the fatty acids themselves. And crucially, they can cross the blood-brain barrier and become a vital, even preferred fuel for the brain during these low-glucose states. It's a fascinating backup system, an adaptive mechanism that ensures continuous brain function even when glucose is perilously low. It really highlights the body's metabolic flexibility and its absolute priority for keeping the brain fueled.
So if fats are going to be burned for fuel in, say, muscle or liver, they need to get to the cellular powerhouses, the mitochondria. How do fatty acids travel from storage in adipose tissue to the cells that need them? And then once inside the cell, how do they cross into that mitochondrial furnace?
Good question. Long-chain fatty acids released from adipose tissue, where they're stored as triacylglycerols, are highly insoluble in the blood plasma. So they travel bound non-covalently to albumin, which is the major protein in blood serum. Albumin acts as their soluble carrier, ferrying them safely through the bloodstream.
Okay, albumin taxis them around.
Pretty much. Upon reaching a target cell, like a muscle cell, they need to get inside. This happens either by passive diffusion for maybe smaller ones, or more commonly and efficiently, via specific fatty acid transport proteins embedded in the cell membrane which facilitate their uptake.
Once inside the cell cytosol, they can't just jump straight into the mitochondria, can they? Let's unpack this. There's an activation step involved first, right, to get them ready.
Yes, absolutely. Before they can be oxidized for energy, fatty acids must first be activated. This means converting them to fatty acetyl-CoA. This crucial reaction takes place mainly on the outer mitochondrial membrane, but also in the endoplasmic reticulum and peroxisomes. It requires ATP for energy, coenzyme A, and is catalyzed by a family of enzymes called acetyl-CoA synthetases, sometimes called thiokinases.
So it costs a bit of energy to get started.
It does. Essentially, a high-energy thioester bond is formed between the fatty acid and coenzyme A using the energy from ATP hydrolysis. This activated fatty acyl-CoA is now primed or ready to burn. It's now poised for its next critical step: transport into the mitochondrial matrix, which is where the main oxidation machinery, beta-oxidation, resides.
That's just to get it ready, activate it, to actually get into the mitochondrial matrix, especially for those long-chain fatty acids. There's a specialized transport system, isn't there? A kind of gatekeeper.
That's precisely where the carnitine shuttle system comes into play. It's a truly vital mechanism for transporting long-chain fatty acids, typically those with 12 to 20 carbons, across the inner mitochondrial membrane, which is impermeable to fatty acyl-CoA itself.
The carnitine shuttle. Okay, how does that work?
It involves three key components. First, there's an enzyme called carnitine palmitoyltransferase I, or CPT I, located on the outer mitochondrial membrane. CPT I transfers the fatty acyl group from CoA onto carnitine, forming fatty acylcarnitine.
So it swaps partners.
Exactly. Think of carnitine as a ferry boat, and CPTI loads the fatty acid cargo onto it. Second, there's the carnitine acylcarnitine translocase, which is embedded in the inner mitochondrial membrane. This protein acts like the ferry dock, facilitating the movement of this fatty acylcarnitine ferry into the mitochondrial matrix, while simultaneously moving free carnitine back out.
A swap across the membrane.
A swap, yes. Finally, once inside the matrix, a second enzyme, carnitine palmitoyltransferase II, or CPTII, transfers the fatty acyl group back from carnitine to mitochondrial coenzyme A. This regenerates fatty acyl-CoA inside the matrix, ready for beta-oxidation, and frees up carnitine to be shuttled back out and pick up another fatty acid. This system is absolutely essential. Without it, long-chain fatty acids simply cannot enter the mitochondrial matrix for oxidation, no matter how much energy the cell needs.
And when this carnitine shuttle system doesn't work correctly, maybe due to a genetic defect, what are some of the severe consequences? What problem does the body face then?
Right. Inherited defects in carnitine metabolism, like deficiencies in the carnitine transporter itself or in the enzyme CPTI or CPTII, can have truly severe clinical consequences. They essentially block the primary highway for cells to access energy from long-chain fats. Patients with CPTI or CPTII deficiency often experience recurrent episodes of severe muscle pain, weakness, and something called myoglobinuria. That's muscle protein breaking down and appearing in the urine, especially during prolonged exercise or fasting.
Because their muscles can't get the fuel they need.
Exactly. Their muscles cannot effectively oxidize long-chain fatty acids for energy during these high-demand periods. Metabolically, they typically present with hypoglycemia, low blood glucose, because they can't spare glucose, and notably, hypoketosis.
Hypoketosis, low ketones.
Yes, a reduced ability to produce ketone bodies, because the fatty acids aren't getting into the mitochondria to be broken down into the acetyl-CoA needed for ketogenesis. It powerfully highlights the absolute necessity of this carnitine shuttle for using long-chain fatty acids, especially when glucose is scarce and fat should be the primary fuel. Imagine having a full tank of gas but absolutely no fuel line to the engine. That's kind of what these patients experience metabolically.
Wow, that paints a clear picture. Okay, so the fatty acyl-CoA makes it into the matrix via the carnitine shuttle. What happens next? How is the energy actually extracted?
Once fatty acyl-CoA is successfully transported into the mitochondrial matrix, it then undergoes the systematic series of 4 recurring reactions known as the Curran oxidation spiral.
Beta-oxidation,
Yeah.
the core process.
The core process. It systematically dismantles the fatty acid chain, 2 carbons at a time, starting from the beta carbon, the second carbon from the CoA end. In each complete cycle of these 4 reactions, oxidation, hydration, another oxidation, and thiolysis cleavage, the fatty acyl-CoA chain is shortened by 2 carbons. And importantly, each cycle produces 1 molecule of acetyl-CoA, one molecule of reduced flavin adenine dinucleotide, FADH2, and one molecule of reduced nicotinamide adenine dinucleotide, NADH.
Okay, so 2 carbons off plus acetyl-CoA, FADH2, and NADH.
Exactly. You can visualize it like a spiral staircase. With each step or turn of the spiral, 2 carbons are cleaved off the fatty acid chain, like removing building blocks. And these valuable energy carriers, FADH2 and NADH, are generated, ready to go to the electron transport chain.
So it's like we're systematically chipping away at the fatty acid chain, getting these smaller energy packets each time. And what happens to all those FADH2, NADH, and acetyl-CoA molecules produced at each turn of the spiral? Where do they go to make ATP?
Right, the payoff. The FADH2 and NADH molecules generated directly feed into the electron transport chain, which is located right there on the inner mitochondrial membrane. Here, they donate their high-energy electrons, setting off that cascade that ultimately drives the pumping of protons and the synthesis of large amounts of ATP through oxidative phosphorylation. So they're direct contributors to ATP production.
Okay, straight to the power generator.
Pretty much. The acetyl-CoA molecules, on the other hand, enter the tricarboxylic acid TCA cycle, also known as the Krebs cycle, which also takes place within the mitochondrial matrix. In the TCA cycle, the 2 carbons from acetyl-CoA are further oxidized completely to carbon dioxide, CO2, producing even more ATP indirectly, along with additional NADH and FADH2 that also feed into the electron transport chain.
So acetyl-CoA yields even more energy via the TCA cycle.
Yes, it's a highly efficient, multi-stage energy-generating process designed to extract the maximum possible energy from each fatty acid molecule.
You mentioned efficiency. Can you give us a sense of the energy yield? How much ATP are we talking about from, say, a common fatty acid?
Sure. To give you a concrete sense of the sheer energy yield, let's consider the complete oxidation of one molecule of palmitoyl-CoA. Palmitate is a very common 16-carbon saturated fatty acid. This single molecule undergoes 7 full rounds of back oxidation. This yields 8 molecules of acetyl-CoA, because the last round splits a 4-carbon chain into 2 acetyl-CoAs, 7 FADH2, and 7 NADH.
Okay, 8, 7, and 7.
Right. Now, when you calculate the ATP equivalents generated from all these products running the 8 acetyl-CoA through the TCA cycle and the 7 FADH2 and 7 NADH through the electron transport chain, it ultimately translates to approximately 108 molecules of ATP, maybe slightly more or less depending on the exact shuttle mechanisms used, but around that number.
108 ATP from one 16-carbon fatty acid? That's huge compared to glucose.
It is a remarkable amount of energy from a single fatty acid molecule. It really showcases the incredible efficiency and energy density of fat as a fuel source compared to, say, glucose, which yields maybe 32 ATP. It's why we can store so much energy in a relatively compact form in adipose tissue.
That's an astonishing amount. But, not all fatty acids are neat 16-carbon saturated chains. They come in different lengths and saturation levels with double bonds. Does beta-oxidation handle them all the same way, or are there specialized enzymes or steps for these variations?
Not quite the same way. There's a fascinating level of specialization involved, adapting the core process. There's significant chain length specificity among the first enzymes, the acetyl-CoA dehydrogenases. For example, there's very long chain acetyl-CoA dehydrogenase, VLACD, which handles fatty acids with maybe 14-20 carbons. Then medium-chain acetyl-CoA dehydrogenase, MCAD, processes those with 6-12 carbons, and short-chain acetyl-CoA dehydrogenase, SCAD, works on the really short 4-6 carbon ones.
So different enzymes for different lengths. Makes sense for efficiency.
Right, they're finely tuned. Now, for unsaturated fatty acids, the ones with double bonds, additional enzymes are required. You need isomerases to potentially shift the position of a double bond, or reductases to remove a double bond, so that the molecule fits correctly into the standard oxidation enzymes.
So extra steps for the double bonds.
Exactly. And for odd-chain fatty acids, those with an odd number of carbons, which are less common but do recur, the final oxidation cycle produces a 3-carbon molecule called propionyl-CoA instead of the usual 2-carbon acetyl-CoA. This propionyl-CoA then undergoes a separate multi-step pathway. This notably requires vitamin B12 as a cofactor, and it gets converted eventually to succinyl-CoA, which is an intermediate of the TCA cycle.
Ah, so even the odd ones get fed back into the main energy cycle.
Precisely. It demonstrates how even unusual fatty acids are fully integrated into the body's central energy metabolism. The system is remarkably adaptable. #tag alternative oxidation routes.
So mitochondrial beta-oxidation is clearly the main highway for breaking down most fats. But are there any sort of alternate routes or side roads for fatty acid oxidation? Maybe for special cases like those very long chains you mentioned or unusual structures. What if a fatty acid is just too long or branched for the main pathway?
Yes, there are indeed crucial alternative routes. These are essential for specific types of fatty acids or under certain conditions where the mitochondria can't handle them. One really important alternative is peroxisomal oxidation. Peroxisomes are another type of organelle in the cell.
Peroxisomes.
This pathway primarily handles the very long-chain fatty acids, typically those longer than 20 carbons, and also branched-chain fatty acids like phytanic acid, which we get from dairy and some meats, which cannot be efficiently processed by the mitochondrial oxidation machinery. While the cycle is similar chemically, 4 steps, removing 2 carbons at a time, peroxisomal oxidation has a key difference in the first step. It generates hydrogen peroxide, H2O2, a reactive oxygen species, instead of directly producing FADH2 and ATP.
Hydrogen peroxide. Isn't that dangerous?
It can be, yes. But peroxisomes are equipped with high levels of the enzyme catalase, which immediately breaks down the H2O2 into harmless water and oxygen. So it's safely detoxified right there. Another alternative pathway is omega oxidation. This occurs mainly in the endoplasmic reticulum and involves cytochrome P450 enzymes.
Omega oxidation, what does that target?
It oxidizes the methyl end, the omega carbon, the carbon furthest from the carboxyl group of the fatty acid. This typically produces dicarboxylic acids, acids with carboxyl groups at both ends, that are more water-soluble and can then be excreted in the urine. These pathways are kind of like specialized cleanup crews for fats that the main mitochondrial system can't easily handle, ensuring no fatty acid goes unused or accumulates to toxic levels.
And presumably these alternative pathways can have pretty significant clinical implications if they don't function correctly. What happens when these cleanup crews are deficient?
Absolutely. The proper functioning of these alternative pathways is critical, and genetic disorders affecting them can be devastating, really highlighting their necessity. A prime example is Zellweger syndrome. This is a severe genetic disorder characterized by a defect in peroxisomal biogenesis. The body literally cannot form functional peroxisomes.
No peroxisomes.
Or severely dysfunctional ones. This leads to the accumulation of very long-chain fatty acids and other things that peroxisomes normally handle, like branched-chain fatty acids. These accumulate to toxic levels in tissues. The consequences are severe developmental and neurological problems, affecting multiple organ systems, including the liver, brain, and kidneys. Sadly, it often leads to early death. It powerfully highlights the critical importance of these less common pathways for handling specific lipid types and preventing toxic accumulation. Without these specialized systems, the body can quickly become overwhelmed, leading to profound impacts on overall health and development.
You mentioned earlier, quite fascinatingly, that the brain can use ketone bodies during prolonged fasting. What exactly are these ketone bodies and how are they produced, primarily by the liver, as this sort of emergency fuel?
Right. Ketone bodies are a fascinating group of molecules. They are water-soluble, unlike most lipids, and there are 3 main ones: acetoacetate or hydroxybutyrate and acetone. They are produced primarily in the liver mitochondria through a process called ketogenesis. This process ramps up significantly when fatty acid oxidation is running at a very high rate and carbohydrate stores and thus glucose availability are low.
So lots of fat burning, not much sugar.
Exactly. Think prolonged fasting, starvation, or crucially, in uncontrolled type 1 diabetes where insulin isn't working. The process starts when the liver is producing lots of acetyl-CoA from beta-oxidation, more than the TCA cycle can handle. 2 acetyl-CoA molecules condense to form acetoacetyl-CoA. This then undergoes further reactions to yield acetoacetate. Acetoacetate can then be reduced to feed hydroxybutyrate, which is actually the most abundant ketone body circulating in the blood during ketosis.
Okay, acetoacetate and beta-hydroxybutyrate. What about acetone?
Acetone is kind of a volatile byproduct. It's formed by the spontaneous, non-enzymatic decarboxylation, loss of CO2, of acetoacetate. It doesn't really get used for energy, but it gets breathed out. It's what gives ketotic breath its distinctive, often described as fruity or nail polish remover-like smell, especially noticeable in individuals with diabetic ketoacidosis. It's literally the body's emergency fuel system kicking into high gear, especially for the brain.
So the liver makes them, cranks them out, but doesn't really use them itself. That seems a bit counterintuitive. Where do they go then, and why doesn't the liver just consume its own product?
Exactly, that's a key point. The liver synthesizes ketone bodies but uniquely lacks a critical enzyme called thiophorus, also known as succinyl-CoA.acetoacetate-CoA transferase.
Thiophorus. And that does what?
Thiophorus is essential for activating acetoacetate back to acetoacetyl-CoA so it can be broken down into acetyl-CoA and used for energy. Since the liver lacks this enzyme, it acts as a net producer and exporter of ketone bodies. It makes them but ships them out.
Okay, so it exports them into the blood. Where do they go?
Once synthesized, ketone bodies are released into the blood and transported to peripheral tissues like skeletal muscle, cardiac muscle, and importantly, the brain, especially during prolonged fasting or starvation. These tissues do possess thiophorease and the other necessary enzymes. This allows them to readily take up ketone bodies from the blood and oxidize them for fuel, converting them back to acetyl-CoA, which can then enter their own TCA cycle to generate ATP.
So the liver makes them for other tissues to use.
Precisely, this allows the liver to conserve its own resources, and importantly, to continue performing gluconeogenesis to produce glucose for tissues that absolutely depend on it, like red blood cells, while providing an alternative fuel for the brain and muscles.
And speaking of red blood cells, why can't they use ketone bodies for energy even when they're plentiful in the bloodstream during fasting? They need glucose, right?
Right. Red blood cells are quite specialized. Famously, they lack mitochondria.
No mitochondria.
No mitochondria. And mitochondria are the cellular organelles where both the enzymes involved in ketone body oxidation and the entire TCA cycle reside. Since these critical metabolic pathways occur exclusively within mitochondria, red blood cells simply do not have the cellular machinery required to utilize ketone bodies as a fuel source. They rely solely on anaerobic glycolysis for their ATP production, which is less efficient but crucially mitochondria independent. It's a fundamental limitation tied to their unique job of carrying oxygen.
This whole system of fatty acid oxidation, breaking down fats for energy, is clearly vital for our energy balance. How is it so precisely regulated? It needs to respond to the body's constantly changing energy needs, from just having eaten a big meal to being in a prolonged fast.
The regulation of oxidation is incredibly tightly controlled, yes. It's a true marvel of metabolic engineering, primarily driven by hormonal signals and the availability of substrates, mainly fatty acids. During fasting, for instance, glucagon levels rise significantly while insulin levels fall. This hormonal shift is a major signal.
Glucagon up, insulin down.
Right. This promotes the breakdown of stored triacylglycerols in adipose tissue, lipolysis, releasing fatty acids into the blood. This dramatically increases their availability for oxidation in tissues like the liver and muscles. So, substrate supply is one key factor. At a cellular level, a really key regulatory point is the entry of fatty acids into the mitochondria, controlled by that carnitine shuttle, specifically the CPT I enzyme.
The gatekeeper enzyme.
Exactly, and CPT I is inhibited by a molecule called malonyl-CoA. What's fascinating is that malonyl-CoA is the committed intermediate in fatty acid synthesis.
So the first step of making fat blocks the first step of burning fat.
Precisely. Malonyl-CoA acts as an allosteric inhibitor of CPT I. This inhibition is absolutely crucial because it effectively prevents the simultaneous processes of fatty acid synthesis, which happens when energy is high and malonyl-CoA is being made, and fatty acid oxidation, which needs to happen when energy is low. It's a beautiful example of reciprocal regulation, ensuring the cell isn't wastefully trying to build and burn fat at the exact same time.
That makes perfect sense. And how is malonyl-CoA itself regulated?
Malonyl-CoA's own levels are tightly controlled, primarily by the enzyme that makes it, acetyl-CoA carboxylase, ACC. ACC activity is regulated by phosphorylation. When cellular energy levels are low, meaning high AMP levels, an enzyme called AMP-activated protein kinase, AMPK, gets activated. AMPK phosphorylates and inactivates ACC. This lowers malonyl-CoA levels, which relieves the inhibition on CPT I, thus allowing fatty acid oxidation to increase.
So low energy turns on fat burning.
Essentially, yes. AMPK acts as a cellular energy sensor coordinating these pathways. It's a really sophisticated system.
So what does this all mean in terms of our overall fuel homeostasis? How does the body gracefully switch between burning carbohydrates primarily and then burning fats when needed, depending on our nutritional state?
This metabolic switch is absolutely fundamental to overall fuel homeostasis and our body's adaptation. It ensures our survival under varied conditions. When glucose is abundant, like right after a carbohydrate-rich meal, high insulin levels promote glucose uptake and utilization by tissues. Insulin also stimulates glycogen storage and fat synthesis storage in adipose tissue. In this fed state, fatty acid oxidation is suppressed, partly due to that malonyl-CoA inhibition.
So store energy when it's plentiful.
Right. During fasting or prolonged exercise, however, as blood glucose levels start to drop, glucagon levels rise and insulin falls. This signals the mobilization of fatty acids from adipose tissue into the bloodstream. These fatty acids then become the primary fuel source for many tissues, like muscle and heart, sparing the remaining glucose. Simultaneously, the liver ramps up ketone body production from these fatty acids specifically to feed the brain, which can no longer rely solely on glucose. This adaptive shift ensures a continuous and adequate energy supply to all vital organs, particularly the brain, under varying physiological conditions. It's the body's brilliant strategy for managing its diverse fuel reserves effectively.
Let's look at Lola B, the 16-year-old mentioned with MCAD deficiency. Her case really seems to illustrate what happens when just one of these specific oxidation enzymes, even a seemingly small cog in the machine, is missing.
Yes, Lullaby's history, recurrent episodes of profound fatigue, vomiting, impaired respiration, especially triggered after fasting for more than about 8 hours, points directly to a deficiency in medium-chain acetyl-CoA dehydrogenase, or MCAD.
MCAD deficiency. That affects medium-chain fats.
Exactly. It's actually one of the most common inherited metabolic disorders. It prevents the efficient oxidation of medium-chain fatty acids, those with 6 to 12 carbons. When glucose stores get depleted during fasting, her body cannot effectively access sufficient energy from these medium-chain fats because they accumulate instead of being broken down properly. This leads to severe hypoglycemia because glucose can't be spared by burning fats effectively, and notably hypoketosis, low ketone levels, because ketone bodies can't be produced efficiently from the accumulating medium-chain fatty acids either.
So a double whammy,
Yeah.
low sugar and low ketones.
Exactly. It's a classic example of how a defect in just one specific enzyme within the oxidation pathway can profoundly disrupt the entire energy balance, particularly during periods of metabolic stress like fasting, where fat utilization is absolutely critical. It essentially leaves the body starved of fuel, even when fat reserves are technically available.
And then we have Diane A., the young woman with type 1 diabetes who experienced ketoacidosis. This sounds like the opposite problem, an overproduction issue rather than a deficiency. What's fundamentally going wrong there?
That's precisely correct. In type 1 diabetes, the fundamental issue is a severe lack of insulin production by the pancreas. Without insulin, the body effectively perceives itself to be in a state of starvation, even if there's plenty of glucose floating around in the blood because the cells, especially muscle and fat, can't take up that glucose effectively.
So the body thinks it's starving, even when it's not.
Right. This lack of insulin signal leads to unchecked fat mobilization from adipose tissue and massively increased fatty acid oxidation in the liver. The liver is just flooded with fatty acids. It oxidizes these fatty acids to produce huge amounts of acetyl-CoA. This completely overwhelms the capacity of the liver's TCA cycle, meaning it simply can't process all the incoming acetyl-CoA that way.
Right.
So the excess acetyl-CoA gets shunted massively towards the production of ketone bodies, acetoacetate, and bihydroxybutyrate.
Way too many ketones are made.
Way too many. When these ketone bodies accumulate in the blood much faster than peripheral tissues can possibly use them, their acidic nature causes the blood pH to drop dangerously low. This leads to diabetic ketoacidosis, or DKA, a life-threatening metabolic crisis. It's a truly vivid demonstration of how profoundly disrupted hormonal regulation, in this case, a severe insulin deficiency, can lead to metabolic catastrophe. It really underscores the immense importance of insulin as the master regulator of fuel utilization and storage.
And finally, Otto S.'s situation, using carnitine supplementation to try and improve athletic performance. This points to how even maybe more subtle issues in carnitine levels might impact energy, especially for athletes.
Yes, Otto S is a good example, exploring the idea that maintaining adequate carnitine levels might be vital for maximizing fat oxidation and sustained energy output, particularly for athletes engaging in prolonged activities. While it's not necessarily implying he had a severe deficiency, the idea behind supplementation is that it could potentially support the carnitine shuttle system, maybe optimizing the transport rate of long-chain fatty acids into mitochondria during moderate-intensity exercise.
Making the shuttle more efficient.
Potentially, yeah. By ensuring a more efficient and sustained supply of fatty acids for beta-oxidation, the thinking is it could increase ATP yield and potentially improve endurance, perhaps by delaying the depletion of muscle glycogen stores. While the evidence for performance enhancement in healthy individuals is debated, it highlights the theoretical importance of every component in the fatty acid oxidation pathway, even seemingly subtle aspects like substrate or cofactor availability, for maximizing the body's ability to generate energy from fats. It's about trying to fine-tune that metabolic engine for peak performance.
Okay, so we've thoroughly covered breaking down fats for energy. But what about the other side of the coin, making them? Can our bodies synthesize new fatty acids? And if so, from what primary source? It's not just from dietary fat, right?
Absolutely. Our bodies are remarkably versatile metabolically, capable of building as well as breaking down. We can indeed synthesize new fatty acids through a process called de novo lipogenesis, meaning making new fat. In humans, this occurs predominantly in the liver, but also significantly in adipose tissue and during lactation in the mammary glands as well. The primary precursor, the main starting material for fatty acid synthesis, isn't fat itself, but rather dietary glucose and other carbohydrates.
So carbs get turned into fat.
Essentially, yes. When we ingest excess calories, particularly from carbs that aren't immediately needed for energy or for filling up our glycogen stores, our bodies are very adept at converting that surplus carbohydrate into fatty acids for long-term energy storage. This process occurs mainly in the cytosol of the cell, which is importantly distinct from the mitochondria where beta-oxidation, fat burning, happens, keeps the processes separate.
Okay, cytosol for building, mitochondria for burning.
Right. And the initial building block, the 2-carbon unit used for fatty acid synthesis, is acetyl-CoA, just like it's the end product of beta-oxidation.
Okay, let's unpack this fascinating journey then. How does glucose, a carbohydrate, ultimately get converted into acetyl-CoA in the cytosol, ready to be built into a fatty acid? It sounds like quite an intricate journey from a sugar molecule in the mitochondria to acetyl-CoA out in the cytosol.
It is quite an intricate journey, yes. A brilliant example of how the body cleverly repurposes metabolic intermediates. So glucose is first metabolized through glycolysis out in the cytosol to pyruvate. Pyruvate then enters the mitochondria. Inside the mitochondria, it's converted to acetyl-CoA by the pyruvate dehydrogenase complex.
Okay, so acetyl-CoA is made inside the mitochondria.
Correct. Now, here's the problem. This mitochondrial acetyl-CoA cannot directly cross the inner mitochondrial membrane to get out into the cytosol where fatty acid synthesis needs to happen. It's effectively trapped inside.
It can't get out. So how does it get out?
Instead of acetyl-CoA itself leaving, it combines with oxaloacetate, or OAA, an intermediate of the TCA cycle right there in the mitochondria to form citrate. Citrate is another TCA cycle intermediate. Crucially, citrate can be transported out of the mitochondria into the cytosol via a specific transporter.
Citrate is the escape molecule.
Citrate is the shuttle molecule, yes. Once out in the cytosol, citrate is cleaved by an enzyme called ATP citrate lyase. This cleavage reaction requires ATP and regenerates the acetyl-CoA we need for fatty acid synthesis, plus OAA, now out in the cytosol.
Okay, so the acetyl-CoA is now in the cytosol. What happens to the OAA?
The cytosolic OAA is then usually converted back to malate, and then malate can be oxidatively decarboxylated back to pyruvate by an enzyme called malic enzyme. This last step is particularly neat because it generates NADPH.
NADPH, why is that important?
NADPH is the specific reducing power, the electron donor, that is absolutely required for the reductive steps in fatty acid synthesis. So this shuttle not only gets acetyl-CoA out, but it also helps generate the NADPH needed to build the fat.
Wow, that's efficient.
It is. This pyruvate can then re-enter the mitochondria, completing the cycle. So you can envision this as a sophisticated shuttle system, effectively moving acetyl-CoA building blocks from the mitochondrial matrix to the cytosolic construction site, and conveniently supplying a key energy cofactor along the way.
So we finally have cytosolic acetyl-CoA. What's the very first really critical step in actually starting to build a new fatty acid chain? The committed step, the point of no return.
The very first and most critical step, yes, the rate-limiting and committed step in fatty acid synthesis, is the carboxylation of acetyl-CoA to form malonyl-CoA.
Acetyl-CoA to malonyl-CoA.
Right. This reaction is catalyzed by a crucial enzyme called acetyl-CoA carboxylase, or ACC. This reaction is essentially irreversible under physiological conditions. It requires ATP for energy, carbon dioxide, usually provided as bicarbonate for the added carboxyl group, and biotin as a coenzyme. Biotin acts as a carrier for that activated CO2.
So adding a carb using biotin and ATP.
Exactly. You can picture it as adding a CO2 molecule to the 2-carbon acetyl-CoA to create malonyl-CoA, which is an activated 3-carbon intermediate. The formation of malonyl-CoA is the bottleneck. It determines the overall rate of fatty acid synthesis. This makes ACC the crucial point of regulation, the master switch for turning fat synthesis on or off.
And then comes the really massive enzyme complex that actually constructs the fatty acid chain, the fatty acid synthase, or FAS.
What makes this enzyme so special? It sounds almost like a molecular assembly line.
The fatty acid synthase complex is truly remarkable, yes. It's a huge multifunctional enzyme. In humans and other animals, it consists of 2 identical large polypeptide chains, each containing 7 distinct catalytic activities all linked together.
7 enzymes in one. 7 active sites on each chain working together. It also contains a crucial component called the acylar carrier protein, or ACP. The ACP has a flexible arm, a phosphopentothene group, that acts like a swinging arm to carry the growing fatty acid chain between the different active sites. It works precisely like a highly organized molecular assembly line. Acetyl-CoA provides the initial 2 carbons to get started, the primer, but then malonyl-CoA is the donor for all subsequent 2-carbon units.
So malonyl-CoA adds the carbons.
Yes, 2 carbon units are sequentially added, one at a time derived from malonyl-CoA, which releases that CO2 we just added in the process to the growing fatty acid chain attached to the ACP arm. Each 2-carbon addition cycle involves a series of 4 reactions. Condensation, joining the units, reduction, dehydration, removing water, and a second reduction. These reductions specifically use that NADPH generated by pathways like the citrate shuttle and the pentose phosphate pathway.
Okay, a 4-step cycle, repeated.
Repeated 7 times, typically. This process usually continues until a 16-carbon saturated fatty acid, palmitate, is formed. Palmitate is then released from the ACP by a thioesterase activity that's also part of the complex. It's an incredibly efficient, self-contained molecular factory for building fats from simple precursors.
This sounds like an incredibly complex and energy-intensive process.
Right.
It absolutely needs to be tightly controlled, right? You wouldn't want to be making fat when you need energy. What are the key ways fatty acid synthesis is regulated?
Absolutely. Regulation is key to prevent wasteful fat production. It needs to ensure synthesis only occurs when there's a genuine energy surplus, usually after a high-carbohydrate meal. Acetyl-CoA carboxylase, ACC, being that rate-limiting enzyme, is the primary control point. It's regulated by both allosteric mechanisms, molecules binding to it, and by reversible phosphorylation, adding or removing phosphate groups.
Okay, allosteric and phosphorylation.
For allosteric control, citrate, remember, the molecule that signals abundant substrate coming out of the mitochondria. Citrate allosterically activates ACC. It tells the enzyme, hey, building blocks are plentiful.
Makes sense. Citrate means go.
Right. Conversely, long-chain fatty acyl-CoAs, like palmitoyl-CoA, the end product of the pathway, allosterically inhibit ACC. This provides negative feedback, shutting down further synthesis when enough product has been made.
Body says stop.
Exactly. Hormonally, insulin, which signals the fed state and high glucose levels, profoundly promotes fatty acid synthesis. It does this partly by activating ACC, often through promoting its dephosphorylation. Insulin basically says, store this excess energy. Glucagon, on the other hand, which signals a low-energy fasting state, inhibits ACC, typically by promoting its phosphorylation via pathways involving AMPK. Glucagon says, stop storing, start mobilizing.
So it's like this sophisticated metabolic switch controlled by fuel levels and hormones, turning synthesis on when energy is plentiful and turning it off when energy is needed elsewhere. Prevents wasteful cycles.
Precisely. It optimizes energy storage very effectively.
Now, palmitate, that 16-carbon product, is the main output of FAS. But our bodies use fatty acids longer than that, too. Can we make longer ones?
Yes. While palmitate is the primary product of the main fatty acid synthase complex, our bodies can certainly make longer fatty acids. This process is called elongation. Elongation occurs primarily in the endoplasmic reticulum, ER, using separate enzyme systems from the cytosolic FAS. It extends palmitoyl-CoA, C16, usually first to sterol-CoA, which is an 18-carbon saturated fatty acid. It can even produce very long-chain fatty acids, sometimes up to 22 or 24 carbons in length.
Why would we need such long ones?
These longer fatty acids are particularly important in specialized tissues like the brain, where they are crucial components for synthesizing complex lipids like sphingolipids that are vital for nerve cell membranes and myelin sheaths. The elongation pathway involves a sequential addition of 2 carbon units, again using malonyl-CoA as the donor, similar in principle to FAS, but it utilizes different enzymes and occurs within the ER membrane, allowing for this diversity in fatty acid chain length beyond C16.
And then there's desaturation, adding double bonds to make unsaturated fatty acids. But there's a big catch here, right? There's some specific fatty acids we cannot make ourselves, making them essential in our diet. What does that mean for our health?
Right, that's a really fascinating point about desaturation. Humans do possess desaturase enzymes, primarily located in the endoplasmic reticulum, that can introduce double bonds into fatty acid chains, making them unsaturated. Stearate, C18.0, can be desaturated oleate, C18.1, for example. These desaturase reactions require molecular oxygen, NADH or NADPH, and cytochrome B5. However, there's a critical evolutionary limitation in humans and other mammals. We lack the enzymes needed to introduce double bonds beyond carbon-9, counting from the carboxyl end.
We can't put double bonds far down the chain.
We can't put them at the omega-6 or omega-3 positions. This means we are physiologically unable to synthesize two crucial polyunsaturated fatty acids ourselves. Linoleic acid, which is an omega-6 fatty acid, with the double bond 6 carbons from the omega end, and alpha-linolenic acid, an omega-3 fatty acid.
Linoleic and alpha-linolenic, the essential ones.
Exactly. Therefore, these are termed essential fatty acids and must be obtained from the diet, from vegetable oils, nuts, seeds, leafy greens, etc. This isn't just a biochemical detail, it's a profound evolutionary constraint, highlighting how deeply our metabolic health is tied to specific dietary components. They are absolutely vital, not just on their own, but also because they serve as precursors for synthesizing other important lipids, including the eicosanoids we're about to discuss. They're also critical for maintaining cell membrane fluidity, brain development, and regulating inflammation. Without them, serious deficiency symptoms can arise.
Let's talk about a very special class of lipids now, eicosanoids. What are these 20-carbon fatty acid derivatives, and what makes them unique in their function compared to, say, a typical hormone like insulin?
Right. Eicosanoids, the name comes from the Greek word eikosi, meaning 20, because they're derived from 20 carbon fatty acids. They are a really diverse family of biologically pertinent lipids. The most common precursor in humans is arachidonic acid, which is itself often derived from the essential fatty acid linoleic acid. What makes them truly unique is their mode of action. Unlike classic endocrine hormones that travel through the bloodstream to act on distant target organs, eicosanoids typically act as local hormones or autocoids.
Local hormones, meaning they act nearby.
Exactly. They are usually synthesized very rapidly in response to a specific stimulus like injury or infection and exert their effects on cells in their immediate vicinity or sometimes even on the very cell that produced them, autocrine signaling. And crucially, they are then very quickly inactivated. They include several major classes: prostaglandins, thromboxanes, and leukotrienes, and they play critical roles in an incredibly vast array of physiological processes, particularly inflammation, immune responses, blood clotting, pain perception, fever generation, regulating blood pressure, smooth muscle contraction. The list goes on. Their localized action allows for very precise, rapid responses right where they're needed.
That makes sense for things like inflammation or clotting at a wound site. So where does the body get the arachidonic acid needed to make these powerful local hormones? Is it just floating around waiting?
Not usually, no. Arachidonic acid can be obtained directly from the diet if it's present in foods like meat or eggs, or as mentioned, it can be synthesized in the body from the essential omega-6 fatty acid, linoleic acid. But when the body needs to produce eicosanoids, arachidonic acid is not typically found free-floating in the cell. Instead, it's usually stored esterified within membrane phospholipids, literally built into the structure of cell membranes.
Stored in the membrane itself.
Yes. When a specific stimulus arrives, perhaps a bacterial toxin, tissue damage, or a chemical signal like histamine, an enzyme called phospholipase A2 gets activated. This enzyme then specifically cleaves arachidonic acid out of those membrane phospholipids, releasing it into the cytosol. So you can imagine arachidonic acid as the raw material that's carefully stored right where it might be needed and then quickly liberated and immediately routed into different eicosanoid synthetic pathways only when a signal demands it. This allows for rapid, on-demand synthesis.
And there are two main pathways for eicosanoid synthesis, starting from that arachidonic acid, each leading to different types of molecules with often very different effects. Can you walk us through those?
Precisely. Once arachidonic acid is released, it can go down one of two major enzymatic pathways, the cyclooxygenase pathway or the lipoxygenase pathway.
Cyclooxygenase, COX, and lipoxygenase, LOX.
Exactly. The cyclooxygenase pathway is catalyzed by phe-OX enzymes. There are two main forms, COX-1 and COX-2. This pathway leads to the synthesis of prostaglandins like PGE2, PGI2, and thromboxanes like TXA2. Prostaglandins have incredibly diverse functions. They can mediate pain, fever, and inflammation, regulate blood pressure through vasodilation like PGI2 or constriction, influence uterine contractions, protect the stomach lining, COX-1-derived ones, and affect platelet aggregation. PGI2 inhibits it. Thromboxanes, particularly TXA2 made primarily by platelets, are potent vasoconstrictors and strongly promote platelet aggregation, playing a critical role in forming blood clots, especially after vascular injury.
Okay, so COX makes prostaglandins and thromboxanes. What about the other pathway?
The lipoxygenase pathway involves different enzymes, the lipoxygenases like 5-LOX, 12-LOX. This pathway leads to the synthesis of leukotrienes like LTP4, LTC4, LTD4, LTE4, and other related compounds like lipoxins. Leukotrienes are key mediators in allergic reactions and inflammation. LTD4 is a potent chemoattractant for neutrophils, drawing immune cells to sites of inflammation. The cystinoid leukotrienes, LTC4, D4, E4, are powerful bronchoconstrictors. They tighten the airways and increase vascular permeability, playing major roles in asthma and
So
anaphylaxis.
different pathways, different products, different effects, all from arachidonic acid.
Exactly. It demonstrates how different enzyme systems can channel a common precursor into generating a diverse array of potent biological signals tailored for specific situations.
This brings us to a very common clinical application that many of us are familiar with. How do drugs like aspirin and other NSAIDs, non-steroidal anti-inflammatory drugs, like the ibuprofen, work their magic to reduce pain, fever, and inflammation?
Yes, this is a classic example of targeted drug action. Aspirin and NSAIDs like ibuprofen or naproxen exert their main therapeutic effects precisely by targeting and inhibiting the cyclooxygenase COX enzyme.
They block the COX pathway.
They block the COX pathway. Most NSAIDs reversibly inhibit both COX-1 and COX-2. By blocking COX activity, they prevent the synthesis of those prostaglandins that mediate pain signaling, inflammation, and fever generation, thereby providing relief from those symptoms. They also inhibit the production of thromboxanes, particularly TXA2, which explains their antiplatelet effects and why they can increase bleeding risk.
Is aspirin different?
Aspirin is unique because it irreversibly inhibits COX enzymes, particularly COX-1 in platelets. Since platelets lack a nucleus and cannot synthesize new enzymes, a single dose of aspirin can inhibit platelet aggregation for the entire lifespan of those platelets, about 7–10 days. This is why low-dose aspirin is used long-term for preventing heart attacks and strokes. It's a clear and powerful example of targeting a specific biochemical pathway for broad therapeutic benefit, but it also explains their common side effects, like potential stomach ulcers by blocking protective prostaglandins made by COX-1 in the gut, or bleeding issues.
These eicosanoids seem to act very quickly and very locally, as you said. How does the body ensure their effects are brief and don't spread systemically or persist indefinitely?
Right, they are designed for transient, localized action. Their effects must be tightly controlled and rapidly terminated, and indeed they are inactivated remarkably quickly, often within seconds to minutes after their synthesis.
Seconds to minutes, that's fast.
Very fast. This rapid inactivation occurs through a variety of metabolic processes, often involving oxidation, reduction, and even vol-oxidation and O-oxidation pathways, similar to how other fatty acids are broken down. These reactions convert the biologically active eicosanoids into inactive metabolites, which are often then further modified, made more water-soluble, and excreted frequently in the urine. This rapid breakdown is absolutely crucial. Imagine the systemic chaos if potent inflammatory signals or blood clotting promoters persisted throughout the body indefinitely. This rapid inactivation ensures their effects remain localized to the site of production and don't persist longer than necessary, allowing for very precise biological control.
And how do eicosanoids actually communicate with the target cells? What's their precise mechanism of action at a cellular level to cause things like muscle contraction or inflammation?
They exert their diverse effects by binding to specific receptors on the surface of their target cells. Most eicosanoid receptors belong to the large family of G-protein coupled receptors, or GPCR.
GVCRs.
Different eicosanoids bind to different subtypes of GPCRs, and this binding triggers a conformational change in the receptor, which then activates intracellular signaling cascades via associated G proteins. These cascades often involve changes in the levels of intracellular second messengers like cyclic AMP, CMP. Some eicosanoids increase KMP, others decrease it, or they might lead to alterations in intracellular calcium concentrations by releasing calcium from internal stores or opening calcium channels. These changes in second messengers then activate downstream protein kinases or other effector molecules, ultimately leading to the specific physiological responses observed in that particular cell type, whether it's smooth muscle contraction, relaxation, secretion of inflammatory mediators, gene expression changes, or altered platelet activity. It's a classic transmembrane signaling pathway, allowing these local lipid messengers to evoke powerful, targeted responses without needing to enter the cell's interior themselves.
Okay, beyond making signaling molecules like eicosanoids, the body also needs to synthesize triacylglycerols for storage, right? Where and how does that happen?
Yes, beyond just fatty acids and these specialized lipids, our bodies also synthesize triacylglycerols, tags, which are the primary storage form of fat, packed away mainly in adipose tissue but also synthesized in the liver. This synthesis occurs predominantly in the liver and adipose tissue, particularly, again, when we have an abundance of carbohydrates or calories overall. The pathway essentially involves combining a glycerol backbone with 3 fatty acids, It starts with glycerol 3-phosphate. Two fatty acyl-CoAs are added to glycerol 3-phosphate to form phosphatidic acid. The phosphate is then removed to yield diacylglycerol, DAG, and finally a third fatty acyl-CoA is added to DAG to form triacylglycerol.
Where does the glycerol 3-phosphate come from?
The glycerol 3-phosphate backbone can be derived from two main sources. In both liver and adipose tissue, it can come from dihydroxyacetone phosphate, DHAP, which is an intermediate of glycolysis. So glucose metabolism directly feeds into TAG synthesis. Alternatively, the liver, and to a lesser extent adipose tissue, has an enzyme called glycerol kinase, which can directly phosphorylate free glycerol to glycerol 3-phosphate. However, white adipose tissue largely lacks glycerol kinase, meaning it must derive its glycerol 3-phosphate primarily from glucose via glycolysis. This tightly links fat storage in adipocytes to glucose availability.
So the liver isn't just a site for synthesis. It also acts as this vital packaging and export center for fats it makes, right? This is where very low-density lipoproteins, or VLDL, come into play. What makes up a VLDL particle, and why are they so crucial for distributing these newly synthesized fats?
You're absolutely right. The liver is a central hub, not just synthesizing fats from excess carbs, but also efficiently packaging and exporting them to other tissues. VLDL particles are the liver's way of doing this. They are produced in the liver primarily to transport these newly synthesized triacylglycerols, along with some cholesterol and phospholipids, from the liver out to peripheral tissues, especially adipose tissue and muscle, particularly during the fed state or when carbohydrate intake is high.
How are they different from chylomicrons?
Structurally, VLDL particles are quite similar to chylomicrons, which carry dietary fat. They have a hydrophobic core of triacylglycerols and cholesterol esters, surrounded by a monolayer of phospholipids, unesterified cholesterol, and crucial apolipoproteins. The key difference is the main structural protein. While chylomicrons have ApoB48, made only in the intestine, VLDL particles contain apolipoprotein B100, ApoB100. ApoB100 is a much larger protein synthesized in the liver, and it's the defining structural scaffold for VLDL and its downstream products like LDL. And importantly, the assembly of ApoB100 with triacylglycerols in the liver's endoplasmic reticulum also absolutely requires that microsomal triglyceride transfer protein, MTP, the same protein needed for chylomicron assembly in the intestine. Its deficiency causes problems in both pathways.
So MTP is needed for both dietary and liver-made fat transport particles.
Correct. You can visualize the synthesis, processing, and secretion of VLDL as this multi-step journey within the liver cell itself. It begins in the rough endoplasmic reticulum with the synthesis of the ApoB100 protein and the initial loading of lipids onto it via MTP. The nascent VLDL particle then moves through the smooth ER and into the Golgi complex, where it acquires more lipids, particularly the bulk of the triacylglycerols, and matures. Finally, these mature VLDL particles are packaged into secretory vesicles and released from the liver cell into the bloodstream via exocytosis. This whole pathway ensures that the liver can efficiently distribute newly synthesized fats to the rest of the body, preventing excessive fat accumulation, fatty liver, within the liver itself under normal conditions.
And once VLDL is in the bloodstream carrying all this liver-made fat, it doesn't just float around indefinitely either. It undergoes a very similar process to chylomicrons, involving LPL again.
Yeah. Yes, lipoprotein lipase, LPL, is central here too, playing a very similar role as it does for chylomicrons in delivering lipids to peripheral tissues. In the capillaries of tissues like muscle and adipose tissue, LPL, which is again anchored to the endothelium and activated by ApoC, which VLDL acquires from HDL in the blood, hydrolyzes the triacylglycerols within the VLDL particle.
So LPL unloads the fat from VLDL too.
Exactly. This releases free fatty acids that are then readily taken up by these tissues for their energy needs or for storage. As VLDL progressively loses its triacylglycerol cargo, it becomes smaller and denser. It transforms first into an intermediate density lipoprotein, IDL. IDLs can either be taken up by the liver directly or they can be further processed in the circulation. With further triacylglycerol removal, often by hepatic lipase, and the exchange of more components with HDL, like returning apocia and acquiring more cholesterol esters, IDL eventually becomes low-density lipoprotein, LDL.
Ah, so VLDL is the precursor to LDL, the bad cholesterol particle.
That's the main pathway, yes. VLDL, IDL up to LDL. So the metabolism of VLDL is directly linked to the levels of LDL cholesterol in the blood. HDL, again, plays a crucial role throughout this remodeling process by facilitating the exchange of apolipoproteins like ApoC and ApoE and lipids between the particles, influencing their overall fate.
Let's look at the clinical side again. Consider Cora N, who presented with hypertension, heart failure, and importantly, elevated LDL cholesterol and elevated triacylglycerols. What metabolic imbalance does this picture suggest?
Cora N's clinical picture, high LDL and high triglycerides, along with cardiovascular issues, is highly consistent with a condition called familial combined hyperlipidemia, or FCH.
Familial combined hyperlipidemia.
Yes. FCH is actually one of the most common genetic forms of hyperlipidemia, increasing the risk for premature heart disease. It's characterized by elevated levels of both cholesterol, often as LDL, and triacylglycerols, often carried in VLDL. The underlying defect can be complex and variable, but it's often linked to an overproduction of VLDL particles by the liver and potentially impaired clearance of VLDL and/or LDL from the bloodstream. So it directly reflects an imbalance in the liver's regulation of lipoprotein synthesis, packaging, and processing. The liver is essentially putting out too many VLDL particles, leading to this cascade of elevated lipids that significantly increases cardiovascular risk.
We also often see a very strong connection between chronic alcoholism and high VLDL levels, leading to high triglycerides.
Yeah.
What's happening biochemically there that links alcohol consumption specifically to fat synthesis and packaging in the liver?
That's a really important clinical connection. Alcohol, ethanol metabolism occurs primarily in the liver, and it significantly alters the liver's metabolic state. Specifically, it leads to a massive increase in the NADH-NAD+ ratio within the liver cells.
Lots of NADH is
A
produced.
huge amount. This high NADH level has profound effects. It strongly inhibits fatty acid oxidation, basically. The liver stops burning fat because it's swimming in NADH from alcohol breakdown. This creates a metabolic traffic jam for fatty acids.
So the fats aren't burned.
They aren't burned. Instead, these available fatty acids, both from the diet and potentially newly synthesized, are shunted towards triacylglycerol synthesis. The liver starts making more tags. This dramatically increased triacylglycerol synthesis, sometimes combined with an impairment in the liver's ability to properly secrete VLDL particles. Alcohol can also impair protein synthesis and secretion pathways, results in an accumulation of fat within the liver itself, leading to alcoholic fatty liver disease, and also an elevation of VLDL levels in the blood, causing hypertriglyceridemia.
So it's a direct consequence of that altered redox state, the high NADH, messing up the normal balance of lipid metabolism in the liver.
Exactly. It leads to both fat buildup in the liver and high triglycerides circulating in the blood. It's a powerful biochemical explanation for a very common clinical problem associated with chronic heavy drinking.
These cases really drive home the point, don't they? From Lola B's MCAD deficiency impacting fat breakdown to Diane A's diabetic ketoacidosis showing the chaos of hormonal dysregulation, to Cora N's hyperlipidemia and the effects of alcohol and VLDL, they all underscore how critical the balance is between lipid synthesis, storage, transport, and breakdown.
Absolutely. They highlight the profound importance of this intricate balance and its complex regulation. They show how even seemingly subtle shifts in a single enzyme's activity, in hormonal signals like insulin or glucagon, or in the overall metabolic environment influenced by diet or toxins like alcohol, can lead to a wide spectrum of complex and varied clinical presentations. These can range from acute life-threatening crises to chronic conditions that significantly impact long-term health and well-being, particularly cardiovascular health. Hashtag taking the dots and final thoughts.
So we're reflecting on everything we've covered across these 3 chapters. What does this all mean for us? We've journeyed through the entire life cycle of dietary lipids, how they're broken down and meticulously absorbed, how our bodies can actually create them from other nutrients like carbohydrates, how they're carefully transported throughout the body like vital cargo and lipoproteins, how they're rigorously burned piece by piece for energy and beta-oxidation, and even how they form crucial signaling molecules like eicosanoids and the fundamental structural components of our very cells. It's a truly vast and interconnected system.
It really is. If we try to connect all these dots, stepping back to see the bigger picture, you can appreciate that lipid metabolism is far from being a simple, isolated set of reactions. It is a highly integrated and exquisitely regulated system, a true marvel of biochemical engineering that has evolved over millennia. The constant interplay between our dietary intake, the precise coordination of hormonal signals like insulin and glucagon, and the specific activities of literally dozens of enzymes ensures that the body can adapt remarkably well to wildly varying fuel demands, whether you're in a state of feasting right after a large meal, undergoing an overnight fast, or even enduring a state of prolonged starvation.
Adapting to whatever state we're in.
Exactly. Every single step we discussed, from the simple but crucial detergent-like action of bile salts enabling digestion, to the intricate molecular machinery of the carnitine shuttle transporting fats into mitochondria, to the complex multi-enzyme factory of fatty acid synthase building new fats from scratch, each one is a testament to the body's biochemical elegance and its profound capacity for sophisticated energy management. It's a system fundamentally designed for metabolic flexibility and resilience.
And it's become abundantly clear through the clinical examples that when any part of this intricate, beautifully balanced system goes awry, whether it's due to a missing or defective enzyme like an MCAD deficiency, a profound hormonal imbalance like in type 1 diabetes, or even certain long-term lifestyle choices impacting liver function or lipoprotein levels, the consequences can range dramatically. We've seen examples stretching from immediate, often uncomfortable digestive issues like steatorrhea, all the way to severe, life-threatening metabolic crises like diabetic ketoacidosis, or the development of chronic conditions like hyperlipidemia that significantly increase the risk of heart disease, stroke, and other long-term complications. The body's ability to handle fats properly is just foundational to overall health.
Absolutely. And this really highlights that understanding these pathways isn't just an academic exercise for biochemistry students. It's absolutely foundational to understanding the molecular basis of both health and disease. This detailed knowledge is what underpins the development of effective diagnostics, how we identify these metabolic disorders, sometimes even before symptoms appear through newborn screening. And it's crucial for the rational design of therapeutics drugs that intervene specifically to correct these imbalances, whether it's inhibiting fat absorption, lowering cholesterol synthesis, or managing blood sugar to prevent ketoacidosis. Ultimately, this fundamental understanding translates into improving human health and quality of life. Without grasping these biochemical nuts and bolts, we'd be largely flying blind and treating many common and serious diseases.
It really makes you think, though. Given how intricate and generally well-regulated these pathways are, designed for survival and energy storage, why do lipid-related health problems like obesity, high cholesterol, metabolic syndrome seem so prevalent in many parts of the world today? What is it about our modern environment or diets that seems to be pushing this finely tuned system out of balance so frequently? That feels like a question that bridges this biochemistry with public health. That's all the time we have for this deep dive into the fascinating world of lipid metabolism, covering digestion, transport, oxidation, and synthesis based on Mark's Section V. We certainly hope this exploration has given you a clear understanding and maybe even sparked your curiosity further. On behalf of the entire team, thank you so much for joining us on the deep dive. Until next time, keep learning and stay curious.